Thank you, everyone. We're going to start this session. We have an esteemed panel here with us today. Everybody come on and get seated. So this panel is regulatory pathway and applications in drug development for rare disease and case studies. So the purpose for this panel I alluded to yesterday. There's been a lot of recent approvals that, they disseminate into the broader community. There's, misconceptions sometimes about the totality of evidence that get discussed in the broader community. So what better way to address this was to assemble a group of people that could speak to different elements of, recent approvals. Obviously, there are some individuals that can't speak directly to their product, but they can talk through the steps on this panel. But what we wanted to do is address those so that people would get an opportunity to see, and hear examples of the dialogues and conversations, but also the levels of evidence that are needed to be done. So I'm going to start this panel with continuing the conversation we had regarding Reata, and Shannon, if you want to interject anything additional after you hear comments from the agency. We have Emily here, who actually did the review and can provide some background experience, and we'll come back and we'll interview people. And after this, I just want to start that for continuation, and we'll go down the row.
Sure, thanks. So for those that don't know me, I'm Emily Freilich. I'm the director of the Division of Neurology 1 at Cedar. And this is a good continuation since we just heard a lot about omaveloxolone and Reata. And I think Dr. Baracchio also mentioned yesterday some of the considerations that went into determination of substantial evidence of effectiveness for that application. As you heard, we have to determine substantial evidence, and typically it can come from two studies, a single study, or a single study plus confirmatory evidence. And as we also heard, Reata, omaveloxolone did have a positive, adequate, and well-controlled study that won on its primary pre-specified endpoint. But it had some limitations as well. It was a small study. It did not have positive findings on the secondary endpoints. And so it wasn't persuasive enough to stand alone as a single study. And so we really were considering what could serve as confirmatory evidence to support what appeared to be a positive treatment effect in the single pivotal study. And they had presented also some mechanistic data that showed that the drug seemed to be doing what it was purported to do. So some biologic plausibility that the drug was having its effect on NRF2 activation. And the natural history study, which we heard a lot about in the last session, but the benefits of that study, or the positives that allowed us to really rely on it, were what we heard a lot about was the natural history database, right? This FACOMS, where we had individual patient-level data and from a large number of patients that had systematically collected the same mFARS that was the endpoint in the primary, in the open label extension for the pivotal study. And so the other thing that was also compelling was that a lot of the centers were the same, and so we felt confident that the data was collected in a similar fashion. I think there was overlap on a lot of the centers between the natural history study and the open label extension study. And so looking at that data and seeing that you had several years of progression in the natural history data and could compare it to the 3 years of open label extension data on the same endpoint on the mFARS, and we're able to do the propensity matching because they had collected demographic data for baseline characteristics to be able to match on the important prognostic variables.
I mean, this is an exciting time. This is one of the reasons I joined CPATH was, to work for the Act for ALS initiative. It was Klaus, I think door number 3 of our game show offer. And so it was an exciting opportunity to work with I've come to know some very incredible collaborators and individuals who are very passionate about working with rare neurodegenerative diseases. And so this session will focus on our efforts that are surrounding the Act for ALS. We'll start with Jacqueline, who you were introduced before, Emily from NIH, and then Alessio Travacchio for FNH. And they'll be discussing some of the efforts that are going on currently and all the great projects that are being planned in real time, as this is really moving very quickly. And I'll pass it to Jacqueline to open us up.
Great, thank you, and thank you for having me to speak about the Act for ALS and where we are today. So I'm going to focus a little bit more on FDA, but of course this is really a joint effort between FDA and NIH and our two public-private partnership partners. So as many of you know, just to level set, passed in December of 2021, it's Accelerating Access to Critical Therapies for ALS Act. We just usually refer it as the Act for ALS. And it had a couple different sections. Section 2 is really geared towards NIH. We won't spend a lot of time, but really using expanded access to do scientific research and use all the data. And then sections 3, 4, and 5 are really ones that FDA had to focus on in partnership with NIH for the public-private partnership, as well as an ALS and other rare neurodegenerative disease action plan, and then our own Rare Neurodegenerative Disease grant program, and I'll spend a little time on that today, and then eventually there will be a report out from the GAO on what we've accomplished. So one of the things, as I said, we were to implement a public-private partnership, and we did that in September 22 when we engaged Critical Path Institute as a convener, and this has recently been expanded, as you'll hear about today, to include FNH and the new Accelerating Medicines Partnership into ALS. And so we're really, very excited about the synergies that both CPATH and FNIH bring to this effort. We were asked to publish and implement a 5-year action plan, which we did, and my slides will be available. That link is live. And included in that was an ALS scientific strategy. And then what I'll spend some time talking about is our award or grant program to public and private entities for research in this area. And just to give you an idea, what we were asked to do was really award grants that were about developing, diagnosing, mitigating, treating, and curing ALS and other rare neurodegenerative diseases in adults and children. And a pretty broad sort of mandate. It was to characterize those diseases in their natural history, to identify molecular targets of neurodegenerative diseases, and then to increase efficiency and productivity of clinical development, including the use of master protocols, add-on trial designs, and clinical trial networks. Now, I will say that, and you'll see in our grants, it's a little hard in a smaller grant program to start a new clinical trial network, so we've really tried to focus on what we think we can accomplish and get results on in the near term. So we stood up the Act for ALS task force internally, and one of the things we wanted to do was reach out to all of our SMEs within CBER, which is the Center for Biologics, CDER, which is our drug center, CDRH, because this is across FDA, so it also includes our device center, and NCTR. And we asked, where are the biggest opportunities for regulatory science? And what we heard was equivalency studies for clinical outcome assessments. Again, sort of going on that theme of can we make trials more decentralized and more accessible. Natural history studies, again, emphasizing the need to further understand these diseases. Our colleagues from CDRH really wanted to work on the enhancement of devices with brain-computer interfaces, and also to think about what about FDA cleared, approved in vitro diagnostic tests for use in diagnosis or treatment. And then, of course, basic translational research related to pathophysiology is always going to be a priority, something probably NIH will do more of than we will in our grant programs. So this was our FY22 awards. Again, we focused on, as we sort of prioritized, 3 different natural history studies, one in ALS, one in ataxia-telangiectasia, and the third one in myotonic dystrophy type 1. So these are all 4-year studies, natural history studies. Then we did some contracts where we are a little bit more involved, and one of them was on the ALS functional rating scale, which I know we just had a discussion about, but really starting to think about remote use equivalency study, as well as a landscape analysis of computer interface-focused patient preference studies in ALS, and that was the first part, which was in landscape. Now, we did have an award, RFI for 23. We haven't announced the awards for this year. They'll be coming shortly, so stay tuned for that. I'm going to talk a little bit about the contracts and why we did this. So again, you heard about this in the previous talk, but we really wanted to look at whether we could get, to the extent that the ALS FRS may not be ideal, but it's certainly used, it can be then brought and used in a more decentralized way. Can we decrease burden, as you heard, improve efficiency, and think about greater diversity and inclusion? And this is just a description of the tool. I won't go over it. I think you've heard a little bit about that. And so, oops, back one, sorry. There we go. So the objectives is really to try and assess comparability of remote use scores compared to in-person scores and assess equivalency between a remote use tool administered via audio platform versus internet. So we have 4 tasks, 2 of which have been completed. One was the landscape analysis of remote use, and a manuscript will be coming out on what we found on that, as well as a pilot study of 12 semi-structured interviews, and that's been done, and that's to identify facilitators and barriers. And so the next part is really doing a feasibility and comparability study at 9 sites geographically dispersed in the US. And included in this is translation and cultural adaptation of the tool to Spanish. We wanted to do that to also make it more accessible. Importantly, as part of this contract, the Office of Administrator really wanted to make sure we had patient input into it as we develop this contract. So we have a patient committee with 3 people with ALS and 3 caregivers to provide ongoing feedback and make sure we're moving in the right direction. Oh, I'm sorry, I keep clicking too much here. The other part we wanted to do was the brain-computer interface, and this is our colleagues in the Center for Devices. And we wanted to see really what's out there, what's the literature about in terms of patient preference studies. Again, these are communication devices, computers, social media. What do we know about what patients think about them? What's their priorities in this area? When we did the literature and landscape analysis, there was not a lot of information, actually. There we go. We're a little sticky today on our... I don't know if you can get me the next slide. I seem to be. Thank you. So there was only really one BCI-focused study that we could find. It was a sample of locked-in patients as well as 13 patients with ALS and their care partners. There was 3-hour home visits and multiple choice. It was an interview. Direct and private forms of communications were the most desired functions, and the only thing the study noted was a little bit of a non-alignment with patients and their caregivers, whereas patients preferred to wait until a BCI was needed, and their caregivers really preferred to have that information a little sooner. So we're going to move on from this, and actually we put out an RFA. So this is really to look at systemic review of clinical outcome assessments for communication brain-computer interface devices. So again, it's what we call this U2, U3 cooperative study, in which the first phase is a systemic landscape analysis, looking at the literature and looking at what's out there in clinical outcome assessments. And then the second phase will consist of patient and caregiver focus groups to collect information about symptoms, functional status, and perceived benefits and risks. So really thinking about how do we move the development of these BCIs forward. There are other, as I said, in 23 we had funding. We had about $5 million funding. What we did was we opened an RFA, an open docket, so that we could understand a little bit of priorities, and we tried to gear that RFA towards what we heard in the open docket. We had 21 comments. Again, improve the understanding of the pathophysiology and advanced biomarker development, things that are probably very familiar to all of you. We are now planning for FY24. We'll be utilizing, again, the concept ideas from stakeholders to develop a new RFA to address unmet needs for neurodegenerative diseases. I just want to also make sure everyone knows is that the Office of Orphan Product Development has a number of grant programs. So this is not limited to rare neurodegenerative diseases. It's their clinical studies of orphan products, and that RFA, the receipt date is in October, but it's every year. There's another one on natural history studies. So although we funded some natural history studies under the Rare Neurodegenerative Diseases, there's other opportunities to have funding for natural history studies. And as I said, the Rare Neurodegenerative Disease RFA for FY24 is in the works. So lots of research that we're trying to promote to inform our regulatory science. This is just contact information. Again, if you have any questions, we're happy to answer them. And I just wanted to really focus on the research and then turn it over to my colleagues in NIH and then our public-private partnerships to talk about the other activities we have. Thank you very much. Okay.
Thank you very much. I'm really glad to be invited to speak during this session. I'm just going to give you an update about NIH activities that are relevant to ALS research. So at the beginning, I just wanted to sort of give a brief synopsis about the disease. As you all know, ALS is a very, very challenging disease. It's an aggressive disease. It causes the rapid degeneration of motor neurons that control voluntary movement as well as breathing. And according to the National ALS Registry, there are at least 32,000 people in the US who live with ALS. And that translates into a lifetime risk of 1 in 400. ALS is characterized by significant clinical and genetic heterogeneity, which of course adds to the challenge in finding treatments. And only about 15% of ALS cases have a family history, which means that the great majority of patients get the disease sporadically, and we really don't understand the causes why these individuals develop ALS. And there are some promising news. By now, we have 4 FDA-approved disease-modifying treatments for ALS. These drugs may have beneficial effects on the disease progression. They may also extend survival. But it's really important to remember, so far there is no known drug that either stops the disease or reverses ALS. So there's a really huge unmet need. And the figure on the right, that is on a graphic that I took from a review article from Pamela Shaw's lab at the University of Sheffield. And I think it really showcases very nicely the biological complexity of ALS. So over the years, many genes have been identified that either cause ALS or are risk factors for the disease. And these gene variants map onto different pathogenic cellular pathways that are either relevant to motor neurons or glial cells, and sometimes also interneurons. And these discoveries are opening many avenues for therapy development in ALS. And I think what is really positive in the field that there's a lot of momentum in ALS research. So currently there are a number of efforts underway to develop gene-targeting treatments for the familial forms of ALS. And in fact, earlier this year, we had an approval by the FDA of a drug called Qalsody, and that is for SOD1-linked ALS. It's relatively rare, but it's an important form of the disease. And parallel efforts are currently underway to also develop gene-targeting strategies for other causative gene variants, including FAS, also C9orf72 repeat expansions, and many more. But what I really wanted to emphasize that there are also new therapeutic targets emerging for the sporadic form of the disease. And these molecules are not only of high interest as therapeutic targets, but also for biomarker development. And I think we heard that in earlier conversations today, how important biomarkers are to optimize drug development in clinical trials in a disease that is as heterogeneous as ALS. This is really particularly important. So last year, NIH led a strategic planning effort for ALS, and we did this through a working group process. And all of these working groups included people with lived experiences in ALS, as well as scientists and clinicians and other subject matter experts. And we had formed 5 working groups that addressed different domains of ALS research. And each of these working groups developed recommendations, which we then pulled together in one strategic plan. And this plan was approved by our advisory council at NIH, or specifically at NINDS, in early February 2023. So we are now in the early implementation phase of our strategic priorities for ALS research. And in fact, on November 6th, NINDS is going to host a community update in which we sort of are going to report out to the community about our efforts to implement these ALS strategic priorities. And we're also going to hear from our nonprofit partners in terms of their recent efforts aimed at advancing ALS research. And the event is going to be fully virtual, so I hope that many of you will be able to tune in. And during this event, we're also going to talk about our efforts to implement the Act for ALS legislation. And of course, this is something that we're doing hand in hand with our partners at the FDA. So I don't think I have to sort of talk a lot about it. So Jacqueline already covered it, but certainly I think the passing of the Act for ALS legislation was a real milestone in the field. And the legislation has multiple provisions, and 2 of them are directly relevant to NIH. And one of them is Section 2, which calls for the development of a grant program for research on expanded access studies in ALS. And the other one is Section 3, and that is the establishment of a public-private partnership in the ALS space, and that is something that we're going to do with the FDA and also many private partners. So this is moving very fast. So in terms of the expanded access program, so this is something that NIH is taking the lead, but I really wanted to emphasize that expanded access is a pathway that is regulated by the FDA. And actually the language here is from the FDA website, so I'm sure, Jacqueline, you're very familiar with it. But so expanded access is also oftentimes referred to as compassionate use. And it is a pathway for a patient with an immediately life-threatening disease, and of course ALS qualifies here, to obtain treatment to an investigational therapy outside a clinical trial in cases when no other satisfactory or comparable alternative treatment option is available. And all of this, I think, is very applicable to ALS. So NIH has released 2 calls for applications, one in the spring of 2022. And based on that call for applications, we are now funding one expanded access study on a compound called trehalose in ALS. And then we had a second call for applications this spring. We've reviewed the application this summer, and we're going to make grant announcements over the next 2 or 3 weeks. So we are approaching the fiscal year, and we're all busy getting those grants ready to give to the investigators to start their studies. And our program really has 2 objectives. One is to conduct scientific research utilizing data from these expanded access studies. And of course, the other aspect is to provide access to investigational therapies for people living with ALS who are no longer eligible to participate in clinical trials. And I think that's a really important issue in ALS, because a large segment of the patient population cannot participate in clinical trials due to the inclusion criteria that are typical for ALS clinical trials. And usually it means you have to be relatively early in the disease to still be eligible to participate in a clinical trial. So a lot of people, they get diagnosed quite slowly, and by the time they really understand what's going on, they are sort of almost no longer eligible to participate in trials. So So this is my last slide, just a few words about the public-private partnership that is being developed. And I wanted to emphasize, and Jacqueline already alluded to that, there are two components of this public-private partnership. So the critical path for rare neurodegenerative diseases was initiated last September, and that's a very close collaboration between the CP-RND Institute, the FDA, and other partners. And in parallel, NIH has been working with the Foundation for NIH to develop an accelerated medicines partnership in ALS. We are currently in the design phase, and here sort of we expect to both analyze and generate large-scale omics data and other biological data in ALS to facilitate and advance biomarker research, but also to identify new therapeutic targets and get a better sense about the causation of sporadic ALS. And what is really central to this effort is the standing up of an ALS knowledge platform. And we expect that this knowledge platform will both include high-quality extant ALS data sets. And actually here we have a working group with the Critical Path Institute and other members from the private sector to identify such data sets. Then, of course, they probably will need to be harmonized, but they will be uploaded into this knowledge platform so that researchers both in academia and industry can access them. But secondly, we are also going to generate new data. And this is going to happen through an ALS research consortium that NIH is intending to fund. We had a call for applications, we reviewed applications, and we are going to, again, make announcements for awardees before the end of the fiscal year. And this research consortium is going to provide a large-scale infrastructure for a prospective longitudinal natural history study in ALS, but also conduct biomarker research, perhaps optimize clinical endpoints, also do expanded access research, and sort of feed into this knowledge platform so that, again, scientists in academia and industry can access the data to advance their own goal. And another really important aspect about that clinical consortium is that they're going to collect longitudinal biospecimen from ALS patients that, again, will be banked in a publicly available repository to provide access to the broader ALS community. And I think this is my last slide, so I think I'll thank you for your attention, and I'll hand off to the next speaker.
Hi, good morning, everyone, and thanks to the organizer for the opportunity. I want to present today the advance in the AMP for ALS, together with a brief introduction on the FNH. So the FNH, or Foundation for the NIH, is not an arm of the NIH, but rather an independent organization funded by Congress to support the mission of the NIH. So basically, when there is the need to support the dollar fund with a PrEP fund from pharmaceutical companies and other organizations, then that's when we step in. Right now, we have over 125 active projects. Over the course of the year, we run over 600 projects and raise over $1.5 billion to support the mission of the NIH. The most famous, I would say, project run by the FNH is ACTIVE, the recent COVID response to develop a therapeutic intervention, a vaccine, done a few years ago. But in the neuroscience space, I would say that ADNI, the Alzheimer's Disease Neuroimaging Initiative, is the most recognized project run by us, together with the AMPs, and I'll tell you a little bit more about that in the next few slides. So what is an AMP? The Accelerated Medicine Partnership started in 2014 when Francis Collins, at the time director of the NIH, was at a HIVR meeting talking with the executive directors and CSO of pharmaceutical companies. And we're discussing why it takes so long to develop a drug. What is the problem? What came up is that industry needs good, strong drug target data, together with good biomarkers to measure target engagement. And that was the inception of the first AMPs, that in the neuroscience space focus on Alzheimer's disease. Now we have AMPs in the neuroscience space focus on Alzheimer's disease, Parkinson's disease, schizophrenia, and now the ALS initiative. Overall, we have 10 AMPs, even though we are planning to develop a few more in the next few years. We raise, through this initiative, we ran over $800 million. We started literally 10 years ago, almost. In February is going to be the anniversary, with 34 industry partners, 16 NIH institutes, and 37 nonprofits working with us. What is AMP-ALS? This is the mission statement that was developed together with the People with Lived Experience and ALS advocacy companies. We'll focus on advanced drug discovery, development, validation of biomarkers and clinical outcome. I'll tell you a little bit more in the next few slides, and new therapeutic drug targets. We are right now in what we call design phase, and we're working very closely together with the NINDS, the FDA, and the Critical Path Institute, together with all our pharma partners and advocacy organizations. And the co-chairs of this initiative are Emily Gubitz, that is here, together with Steph Fredette, that I'm not sure is in the audience right now, but will come probably later today. And then Dr. Off, that is the founder of Target ALS and individual living with ALS. What do we want to do through this initiative? As Emily mentioned before, there are two components of this initiative. There is the harmonization of existing data that is mostly on critical path, and then there is the generation of new data that is mostly on the FNH. And we are working very closely to be sure that we don't reinvent the wheel and we don't duplicate efforts in all this initiative. The first thing that we want to do is harmonize all the existing data. As probably you know, there are many data sets and initiatives focused on ALS. They're all valuable, but if we work all together, it's going to be just much more impactful than each organization can achieve on its own. So the first thing has been identify all these initiatives and all these data sets, and then bringing them all together. And then on the other side, there is the generation of new data through several omics, and this is just some examples of what we can do. We have 5 working groups right now. The first one is the Portal and Data Architecture Working Group. And the goal of this working group is to decide what is the provider of this initiative, what is the portal that we're going to use to not only collect the existing data, but also to include all the other potential omics data that we're going to generate. There is the Extend Data Working Group, and this has been led primarily by Critical Path together with the NINDS to identify existing data sets. And then there is the Biomarker New Clinical Data Work Group, which represents the core of the initiative we can do at the FNH. What are the biomarkers that we need to identify? What is the work that should be done? And since it was mentioned earlier today, I want to add that some of the work that we're doing through this working group, including digital outcome, digital tools that we can use as well. Then we have the Clinical Endpoint Working Group to identify gaps in better stratification of patients and disease phenotyping and phenoconversion. And this is managed by Critical Path together with us and the NINDS. And least but not last, we have a working group focused just on people with lived experience. But again, we have individuals with lived experience in every single working group, including the co-chair of this initiative. And I'll stop with a timeline of this initiative. We started not that long ago, and right now we are in the what we define design phase to define together with the industry partner what are the priorities that we want to work together. We're going to try to wrap this up by mid-October, and then we're going to start with the implementation phase. And the first outcome of this initiative is going to be the establishment of the AMP-ALS portal. That should take about 6 months to be completed, and then a few more months or years to bring in all the data sets and resources that are out there. And I want to add that all this initiative is in the pre-competitive space, so any pharmaceutical companies can access and should access all the data that we will produce. And with this, I'll stop. Happy to take any questions and talk with you now and later. Thank you.
today. I'm going to give the anchor talk, and then we'll open up for questions. I've been fortunate to hear from these, and you'll hear a lot of the similar voices here, because I can tell you, each of us talk, I would say, at least 5 times a week. So it's been quite a bit. Just to remind everyone, Critical Path, our model is built on bringing folks together. I mean, we are a convener, a neutral stakeholder, trusted partner. Our goal is really to identify unmet needs and looking at drug development and bring the best science and consensus to understanding how best to move forward through degeneration, as well as drug development tools. We work closely with regulators to make the non-competitive drug development tools available to the public community so that they can use those to advance therapeutics development. We undergo our submission to all regulatory endorsed with the hope, again, that they can be readily on-ramped into drug development perspectives. Just to give you an idea, a reminder of stakeholders, obviously regulators, the patient and person with lived experience community, nonprofits, industry, and academic researchers are really the cornerstone of these opportunities and efforts that we lead. CP-RND, just to remind everyone, Jacqueline alluded that CPath was selected to establish a public-private partnership with working with the various stakeholders, persons with lived experience, regulators, NIH, FINH. But I want to draw attention, because we hear this a lot, is this only ALS? And it's not. We've been charged with working within the ALS space, but also in the broader community. And so as you hear a lot of discussion in this conversation around ALS, CPath is committed to moving into multiple areas, and I'll talk about those in a minute ago. And we feel that there's great learnings that can be done by engaging other rare neurodegenerative diseases, because some of the questions are similar, and some of the learnings can be shared across different rare diseases. And so when I joined, one of the first initiatives that were done is we moved our ataxia programs at Huntington's under the same umbrella as our CP-RND, largely because they were more established and had learnings that we could draw from. The stakeholder community was well established, and that there are many things that we've discussed with those teams as they were brought on board. Obviously, the ALS initiative, and then we have others that are being brought on through task groups, and you'll hear that throughout the remaining of the time here at this meeting. But think of task groups as sort of directed, specific, early proof of concept areas of focus that CPath undertakes, almost like a bit of a pre-consortia. We have one that's launching right now in PSP. We're targeting one in rare epilepsies and frontotemporal dementias in the beginning part of next year. Obviously, one of the areas that's important to us, and I stress this a lot on our calls, is that we're accountable to the public. So persons with lived experience are critical to this, and they're embedded in all the things that we do. And again, one of the beautiful things about R&D as a model is that we have the shared learning of multiple rare diseases that we can bring to the table to address common questions that we see. I put this slide in as a reminder, because like I said, a lot of the conversations that Alessio and we started about the same time was, how do you all know that you're not duplicating what the other people are doing? And so we kind of came up with some ideas around this, is that, as you heard, there'll be a lot of preclinical and discovery and translational science focus for FINH and NIH, whereas FDA and CPath will be focusing not just on the, some of the review of existing data, but then how do you apply that existing data and new gaps to things that will accelerate questions around regulatory science. One of the things that's integral to this model is the shared connectivity. I'll give you an example. Alessio mentioned the extant data working group. CPath had done landscaping around where all the data sources were. We brought that to the team. We talked about high levels of prioritization, where we could go with it. So the idea is that we can learn and share across functional gaps discussions. And so, our future state will be really looking at, okay, what knowledge gaps that exist, bringing those to stakeholders, be it persons with lived experience, academics, pharma, and then coming up with solutions, either it be through drug development tools or through new RFPs that discover new science. And so this model, we feel is really set, and it sets up a beautiful orchestra from as we've talked about this, because we can have regulatory discussions and the impact of that very early on in the design of NIH's science. So we're excited about that. Now, this is a busy slide. I'm going to go high level of this so we give some time for questions. What I wanted to point out here is a couple points of just what have you been doing in this year. CPATH, if you look at after first quarter, sort of the team that's been here really started in January for the most part. I can tell you we've moved really fast because like Alessio and I started at the end of last year, and we're at the state where we've launched working groups and are ready to roll into scientific plans right now. But what we ended up doing at CPATH is we did a lot of community outreach. We did a lot of trying to understand, where the unmet medical needs were for specific groups in both ALS, but other rare neurodegenerative diseases. We spoke with industry, understanding what their pain points, where did you cry after type B meeting, and things that would really help us say, okay, what can we produce there? We worked in trying to, the ataxia and HD risk program moved to leadership, and they've been working out through a scientific plan, so there's been a lot of restructuring, and there'll be meetings during this time that really flesh that out more detail, but a lot of activities of really re-level setting efforts within the rare neurodegenerative disease at CPATH. We've been actively participating in the AMP for ALS design phase and working on multiple work streams, and then co-leading efforts within the extant data as well as the endpoints and COA working groups. What you're going to see now is in sort of the next phases of this, we're both sort of working up to finish our scientific plans, which we'll be vetting to stakeholders, so that you're going to see towards late this fall and beginning of next year just an explosion of activity around ALS as well as the other neurodegenerative diseases. This just tells you some landscaping things, and I'm not going to go over great detail this, so we give plenty of time for questions, is that, one of the parts that was interesting about CPATH, as the R&D initiative stood up, is that we do have nonprofits and persons with lived experience engaged within other parts of CPATH, but R&D was going to take this to build on this, and we hired Tom Hart on really, and he's built a lot of interactivity and connectivity to the patient's community in ways that we hadn't explored at CPATH. And we're working on now to try to build capacity in this space so that we can really grow interaction and connectivity within patient-informed drug development within R&D and expand that further within the rare disease programs at CPATH. Data landscaping, I can tell you the number of data points we hit for this, no pun intended. We identified 62 plus data sets internationally. We interviewed most of the stakeholders working on data dictionaries, everything from what their architecture is to how many people, what are their biosamples, very great details. Those were summarized and prioritized and shared with the AMP programs so that we could begin to start talking about how do we best align for how they can be incorporated into a larger workspace and data warehouse. COAs, we participate and co-lead that working group. We were fortunate to have people like Michelle Campbell and Charles Moon, really also provide additional advice as we work through this space, because as you heard earlier today, the ALS FRS is posed with some challenges. What we wanted to do is begin to take a larger view of ALS in that, would the concepts around and the measurements that were needed to, as we build capacity and things that impact people at different stages of their disease, as well as symptom domains, were addressed as we begin to expand the level of explosion in therapeutic development in that space. One of the things that CPATH does, if you know us really well, is we do a lot in quantitative modeling. We had our QuantMed people look at existing models and data that was done, so how they could be strengths and weaknesses and applied to regular science, and using AI as well as tools that could be further expanded in that space. And so we've already started looking at, from initial data sets we've had, applying new models and things to them already. Obviously, you've heard about digital health technology. This is one that's very near and dear to my heart, just because I think, one of the things that I heard significantly as we were doing our outreaches was the patient community really wanting access. Access was really stressed, and they felt that that was one of the venues by which they could be accessed. The second part of it is that using DHTs as ways to inform natural history studies so that therapeutic development could be inserted much earlier in conditions. Now our next steps, some of these I'm just going to say to be determined because they're in other sections at this meeting. So the ALS Consortia launch, we're planning finishing up this end of this year, and we're launching into the first January, February 24. We'll be talking about the focus group areas that we're working streams, and we want to hear back from prioritization in the session later today. HD Risk will be talking about their scientific plan, I think, and that does post-meeting discussion, so I won't steal their thunder there. CPTA has been really working on focusing on a staging system. One of the things that's beautiful about staging systems is they help with trial planning, but understanding, and setting the foundational steps for modeling. And they'll also be looking at trying to look at endpoints, specifically earlier in disease progression for the spinocerebellar ataxias. We'll be working a lot to expand the model of patient-formed drug development, and I want to thank those who are tuned in that couldn't make this meeting. I know that your time and investment and attendance is greatly valued, and we're glad you could listen in, and we feel very fortunate because many of you that couldn't be here today have been actively meeting and working with all of us on this stage and others in this audience. And so we're thankful for your time and your commitment to us moving this initiative forward. As I mentioned, we're not done yet. R&D will be actively looking at new areas, so reach out to us. We want to. It's good to have the problem of too much to do, and so one of the things that we really want to hear from the rare neurodegenerative communities is, where do we go next? Because one of the things we were hoping to do is hold a meeting in the first quarter of next year to identify new areas and develop project plans. And this will really work with the connectivity with RDCA-DAP because as you'll see, they'll be incubated in that system and connected to the CP-RND programs. And so we're committed to a research roundtable after that next steps. And I will stop now for questions from the audience. Go back. And before we have questions, I want to acknowledge some folks in the room. For those of you who are working in rare neurodegenerative diseases, can you raise your hand or stand up or something? There's a lot of people in this room. We're all committed to making this better. I can tell you I'm very, very excited about the colleagues that we work with because we're all very passionate. Egos get checked at the door, and we're all just roll up our sleeves and get this thing moving. So we're excited, and next year you're going to be hearing more and more from us as we begin to make momentum in this space, and we're definitely be in touch with it. So questions from the audience.
Hi, Colin. Andrea Pauls-Backman, ALS Strategy Consulting. I've been involved in this space for 10 years. I've never seen this level of cooperation among the various government organizations, the regulators, and I just want to say I'm really thankful for CPATH getting involved, especially in the ALS space. So my question is this. Given that you've done this work in other rare diseases, what has surprised you the most? And you and I have worked together a little bit over the last few months, full disclosure. What has surprised you the most in coming into the ALS space?
For me, I can say for me, I'm fairly new to the ALS space. I can say with the things aren't necessarily dissimilar, and I think we've talked about this in some of in other meetings. But one of the things to me is that there's a lot of activity here, and I think that there's a lot of passion from the individuals here. I think the challenge for us now is really connecting the dots. So like making sure that you're doing this, you're doing this, you're doing this, and you're doing this, well, along with the goal of, you look, pediatric cancers, for those, pediatric cancers excelled because of their willingness to all come together along the same trajectory, and it resulted in really prolonged lifespans for pediatric cancers. And I think that ALS, what I'm seeing now is that there's more stuff, more data and more things here than I think I would have originally said.
But I think now is just the putting it all together and us, kicking the blocks off the tires and getting moving. So, yeah, Emily, were you going to say something?
I mean, the question was for you, but I mean, when I think about ALS, I think one thing that is important to remember that to think globally about ALS. And there are so many excellent efforts in Europe, in Australia, in Japan, and just sort of always. And I think that was sort of one of the surprises when you guys started collating the high quality extant data sets. There are so many international studies and sort of trying to sort of now tap into them. I think it's a huge opportunity, especially since some of the efforts, they're also relatively new there. So we need to connect with investigators, align in terms of what they're measuring, how can we do it in similar interoperable ways. So, I mean, that's always like something that I really want to remind myself. It's a rare disease, it occurs globally, and there's a whole sort of international community.
I want to echo that. That's another thing that's going to be, you're going to hear as this moves out, is that I think the vision of this will be more global. And so, there's, in our minds, we've already been talking to international groups. You're going to hear that in conversations. So others that want to?
Another comment. What surprised me the most is that the field is very fractured, as you know, but every single organization has been so nice and easy to work with. And I asked the question, why is the case? And it seems that this is the time. So as we are working all together, all these foundations are coming together as well. And so this initiative, I really hope that it's going to be successful because literally every foundation has been supportive and collaborative.
Well, thank you. I want to say, I said something to Emily earlier this morning. We're at a place in the ALS landscape where for the very first time, there are funds available, significant funds available for research and for programs like these, and we have the coordination. So those two things are key. Thank you.
Thank you very much. I'm Kathy Collett from Indianapolis, Indiana, a longtime ALS advocate. I love what I hear, and I'm most appreciative, but something is bothering me in the back of my head, so while you're all here, I'm going to tell you what's bothering me. We saw FDA grants for natural history studies, plural. And that's like, I get it, but at the same time, I'm thinking we need a honking big natural history study, and we need to get all these other natural history studies together. And then I hear about harmonizing data. And I think harmonizing is a word that is not well defined. And I think we have a lot of people doing a lot of good ALS research. And when they hear harmonizing, they're like, good, you'll make my data fit with the rest, and you can use it. And I know the word standards is not something that creative people like, but as a part of this, can we come up with some standards so that people are actually generating data at the source that make the data much more combinable? Because today, there's no doubt we have terrible silos. So thank you if you can reassure me.
Do you want to take that with?
Yeah, I mean, certainly, that is something that we think about on a daily basis. There are common data elements for ALS that were developed by the community, by NIH, but it's still sort of a little bit difficult to implement them. But in terms of the natural history study that FDA is now funding and sort of synergizing effort that is moving forward to NIH, we are going to talk, and we are going to talk with the investigators. There'll be overlapping sites, so it's not that we sort of do things in silos. Right. This is sort of more of a continuum. And natural history studies also look a little bit different in terms of what they want to focus on. And there are different outcome measures, and maybe one natural history study will sort of emphasize the collection of longitudinal samples for, high new omics technologies, or another one, they want to go more into the direction of digital outcome measures, things like that. So, but what is really important that we'll be able then to combine the data and sort of make it a stronger, bigger study, exactly. Just up the sample numbers, as long as we ensure that the data is collected in the same way. And I mean, it's a little bit more difficult sort of thinking about older studies that, were performed a few years back. But even then, we already had some data standards, but it's something that is sort of we're thinking about every day. And it's, I think the investigators are very much aware of it.
Yeah, and just to add to that, that's something I think that we're in an opportune time, because sitting in this room, I see a lot of people that do data standardization all of the time, and they make it for multiple uses. And I think that the beauty of this now is that we have the ability to connect this and speak to this from a broader community. And I think if we're going to get there, the time and talent and treasure is all in this ecosystem now to make that happen. And like I said, the other part is feedback from people like you that keep us honest. Okay, last question from the audience here.
Hi, I'm Frank Serwick. I'm Chief Medical Officer of Spiro Pharmaceuticals. We don't do ALS research, but I've been very involved in critical path and other areas for many years and have watched the evolution. And I'm really thrilled and congratulate you on how close you are to the pinnacle of collaboration and cooperation today as compared to where we were when it wasn't Critical Path Institute, but Critical Path Initiatives, one separate for NIH, one separate for FDA, who didn't talk to each other until the patient foundations actually brought that together in a public-private partnership. So the one comment I have or question I have is sort of addressed in terms of the about to collaborate and discuss the work that each agency is funded to do. I would just encourage NIH and FDA, which you've said you already are going to do, even as I was standing up here, to talk and to learn from each other, particularly from the perspective of the experience that we had in autosomal dominant polycystic kidney disease, where there wasn't that communication. And so NIH developed a very expensive and long program to develop an endpoint that they hoped would be useful for drug development. It actually was. It ended up being very helpful, but it took an evolution, and it took that bringing together. So I would encourage you that have an NIH reviewer or someone speak with you about your programs in terms of the clinical research that you're going to be funding, and vice versa, have someone from FDA very involved in the continuing clinical research you're doing here and in other areas, so that there's an understanding of both the basic and practical science that's needed. Thank you.
Well, thank you. Thank you, everyone, for this. For the being able to attend the session. Sound? Can you hear? Okay, thank you. That will conclude this session, and then we will go ahead and pass to the next.